Hi, I'm Anastasia. Today I'm going to demonstrate how to make algae balls for the lab investigating photosynthesis with algae balls from Mass Bioed. One of the solutions for this experiment, sodium alginate, should be prepared at least 24 hours in advance because it takes a long time to dissolve. It is important to use a concentrated algae solution. I'll be using a solution of nanochloropsis, which is a single-celled freshwater algae. This solution has been sitting undisturbed for a few hours. All the algae has sunk to the bottom. I'll be using a 5 milliliter pipette in order to pipette the solution into my container. Place your thumb over the top of the pipette and lower it into the solution. When you release your finger, the solution will be drawn up into the pipette because of negative pressure. Then, you can transfer your solution into the container by removing your thumb. Now I'm going to transfer 1 milliliter of the solution into our tubes. Great. Now you can centrifuge these for 30 seconds at 5,000 to 6,000 RPM. Once the samples are ready, you can pour off the supernatant into a waste container. Then you can actually repeat the step with more algae solution to get it to be more concentrated. After the second centrifugation step, you can pour off the supernatant, but this time resuspend your solution in 0.5 milliliters of the algal culture. After pipetting, your solution should be a dark green. Now I'm going to show you how to make the algae balls. You're going to need to mix an equal amount of algae solution with sodium alginate. So I'm going to add 0.5 milliliters into our solution and pipette by gently mixing up and down. Sodium alginate is very viscous, so it takes a little while for it to go into the pipette. Now I'm pipetting it. Now I'm going to show you how to make the algae balls. Add this mix into calcium chloride by pipetting individual droplets. Let them harden for 5 minutes. After 5 minutes, use a tea strainer or a plastic screen in order to strain the algae balls. Next, you can rinse them with water. Great, your algae balls are now ready to use. I'm just going to spoon them into a new tube. Super. The last step is to make the indicator. We'll be using a 1x hydrocarbon indicator solution to measure photosynthesis. Since the solution needs a high level of carbon dioxide, you can blow through a straw until the color changes to orange. Now you can transfer your indicator to a smaller tube. Finally, you could add the algae balls and the indicator to a tube and you're ready to use it for your experiments. After 20 to 30 minutes, compare the color in your indicator to the color in the standards. This concludes our video for today. I hope you found these tips helpful.